Okay, so good morning and good evening, everyone. Welcome to this time's MU Zoom. So today it's my really great pleasure to welcome my colleague and friend, Ben Afzali from NIDDK, give us actually a COVID talk. I think this is the first time the speaker I invited give about COVID theme to the audience. So it's great. So before we get into the seminar, just to give a brief introduction about Ben. So Ben actually get his MD degree in UK, which he got it in United Medical and Dental School of Guy's and St. Thomas Hospital. And then he pursued his academic career also in UK in King's College in PhD with immunology. So afterwards, I think Ben decided to, actually he interested very much in research, particularly in immunology. So he decided to take a further step to do his postdoc training in NIH, actually in John Xie's lab. And then because he likes NIH so much, so he decided to stay in NIH after he finished his training. And then he started his own lab in NIDDK as a tenure track investigator. So Ben's lab actually is focused on a more kidney disease. So his lab is more interested in studying immunoregulation, how transcription signals from the environment are integrated into immune cells to instruct fate decision and direct effect functions. That's a relatively general goal, but his particular aim is to better understand how the immune regulation is perturbed in disease of autoimmunity that affects kidney, and why resolution of inflammation can be followed either by tissue healing or irreversible scarring. So his lab is also trying to develop new strategies that manipulate the balance between the inflammatory and the regulatory immune response, and to minimize or prevent kidney diseases. So the main theme his lab actually focuses on is one is understanding how transcription signals from the environment are integrated, particularly in T cells, to determine inflammatory versus regulatory T cell differentiation and effect functions. And the second part, his lab also trying to understand how mediators produced by T cells in tissue determine how whether inflammation results in tissue healing or scarring. So Ben really has a stellar CV. I think I'm going to skip the early period of his academic career, but I think since he came to NIH, every single year he got a performance award. That's a quite remarkable achievement. And I think this year he also got NIH nominee for Pew Biomedical Scholars Program. When I look at Ben's publication, it's really impressive. He's been super busy with over 70 different publications in a very short period of time. I'm rather impressed for myself. But without further ado, I think we are all looking forward to Ben's new seminar about COVID-19, the new generation approach to untangling the COVID-19. So without further ado, Ben, please take away.
Thank you so much, Chuan, for that really lovely introduction, and thank you for the invitation to speak tonight, this morning. It's a real pleasure to be here, and I hope this is something that you all find interesting. What I'm going to try and do is just give a very brief sort of outline of how we approach science in my lab, and then talk to you about 3 different stories. And the first story is going to be very quick, just to set the stage. Number 2 is going to be a bit longer, again, to set the stage. And number 3 is all unpublished data, which I hope you'll find interesting. It really illustrates what I think is the power of our approach. So let me just start by saying that so we have only 2 or 3 central questions. Number 1, what are the molecular mechanisms that underlie tissue inflammation? Number 2, how can we actually manipulate these? And the reason for these two questions really is to find ways that we can prevent or ameliorate inflammation in order to preserve healthy tissues. Because when you have inflammation in an organ like the kidneys, the consequence of it is permanent loss of some of that kidney tissue. And that's because we don't know how to prevent the scarring. We also don't know the mechanisms that underlie it. And so early intervention to prevent disease and preserve healthy tissue is really key here. So my lab is really heavily invested in pattern recognition in large and complex data sets, by which we mean high throughput assays involving sequencing. So this is the gamut of the technologies that we use in our lab. So transcriptome profiling, so we're talking about RNA-seq, transcription factor distribution profiling, ChIP-seq, cut-and-run, cut-and-tag, epigenome profiling, again, similar stuff. You know, the chromatin architecture in 3D, CyTOF, and methodologies that validate that, including genome editing. So the way that we approach this is to ask a question, and then instead of approaching it with a hypothesis-driven option, we try and approach it in a discovery-led way. So I'll give you a sort of an example of how we do this. And most of my talk is going to be based on this very simplistic cartoon. So imagine you have a tissue here, and this tissue is a box here. So everything in here is a tissue, and an immune cell has to come from the outside, usually into that tissue at some point. And within that tissue, you'll have a group of inflammatory cells, you have a group of regulatory cells, and the idea is that these things induce inflammation, these things here are anti-inflammatory and cross-regulate those. And at some point, some of these inflammatory cells can become regulatory or contract. Now, generally speaking, for the purposes of this talk, everything that's in red will be towards the inflammatory, will sort of reflect tissue inflammation or infection and injury. And anything in blue is more tissue homeostasis, healing or repair. And of course, within these tissues, we have accessory cells, let's call these non-immune cells. And for the want of a better phrase, let's just consider epithelial cells for the purposes of this talk. So an illustrative example of how we approach this is to ask a simple question, right? So number one, what happens to cells when they enter tissues? So to answer this question, what we did was to take the transcriptomes of cells in tissues and compare them simply to the transcriptomes of cells outside. By looking at these differences, we can see what kind of signals these cells have received in order to express their program. And some of those signals will be received in tissue, some of those will be received potentially as they come into tissues. And sort of to summarize the story, what we found is surprisingly that the cells that come into tissues start making complement proteins. And this was really quite a surprise. And so I think I really have to illustrate this because it really is a surprise. So here is all the pathways that are induced in the cells that are in tissues compared to circulating cells, ranked by the significance of enrichment. So these are all pathways that were enriched. And you can see the number one pathway of cells in tissues is the complement and coagulation cascade. And number two is the same. So this was really quite a surprise, that you find that cells that come into tissues express, complement factors. Let me give you a little sort of background on the perceived wisdom about the complement system. So this is we've all seen diagrams of this. Complement system is supposed to be a major component of innate immunity, and it's effectively a series of proteins and proteases that generate effector molecules sequentially through enzymatic reactions. So in this system, we know that a lot of this is strongly pro-inflammatory, and that it's an essential component of immunity. And this is one of the most ancient and evolutionarily conserved components of the immune system. We know that a lot of the complement system works through interactions between receptors and ligands. And so C3b binds CD4 T, C3a binds C3a receptor, and C5a binds a C5a receptor. We're going to come back to these three things during the course of this talk. We also know that based on this diagram alone, that there are two key effector molecules, C3 and C5. And for the purposes of this talk, we're going to focus mostly on C3. Everyone thinks that this is a serum, in fact, plasma-based system. And that is one of the perceived wisdoms that really has been challenged in the recent past, because we know that complement is also present and active intracellularly in almost every cell that we've tested. So having set the stage, I'm just going to finish the summary of this particular story, and we'll come back to factors B and D a little later, to say that what we found was that cells that are in the periphery receive a set of signals during diapedesis into tissues. And this involves interactions between adhesion molecules expressed on the surface of the cell and adhesion molecules on activated endothelial cells, in this case, LFA1 and ICAM1. That signal transduces signaling through the AP1 complex, which binds the C3 gene and actually licenses the cells to produce C3. Now, what is so important about this? Well, what we discovered in this paper is that the absence of C3 renders T cells unable to produce interferon gamma and become Th1s. So patients who have deficiencies of LFA1, they have a well-characterized immunodeficiency, but we found they do not express intracellular C3. And in fact, if you just replace the C3 with C3 mRNA intracellularly, they start making interferon gamma. So this is a key component of the biology of T cells in tissues. And so you can read all about this story. This was published in Immunity last year. The two key collaborators here are Majid Kazemian at Purdue University and Claudia Kempen at NHLBI. And the first author among some is Martin Kolev. So having set this stage, we asked the next question, which is, okay, so these cells are in the tissues. What are the signals that are derived from tissue cells that are non-immune that influence the biology of these cells? Now, having had access to COVID samples, it occurred to us that COVID is a perfect testing ground for this, because we know that infected cells must be producing some kind of signals. And the summary of this story is that we found that these cells also produce complement factors. And I'm going to just explain this a little bit more detail here. So we performed exactly the same methodology. We compared the transcriptome of infected versus uninfected cells. And we found that if we took to normal human bronchial epithelial cells, infected versus uninfected, A549 cells, these are lung epithelial cells, infected versus uninfected, A549 cells that were first transduced with ACE2, or even patient cells, and comparing again infected versus uninfected. And we looked at all the pathways that are enriched. There are 14 that are in common between all sample types. Of these 14, 4 of them are complement pathways. And to illustrate that, this is the heat map of the leading edge of genes in one of these pathways. So as you can see, C3 that we discussed earlier is one of the top enriched genes. And also CFB, this was complement factor B, which is part of the alternate pathway of activating complement. So this was quite intriguing to us. And what we went and did is to try and figure out how these molecules were induced transcriptionally within infected cells. And what you can see here from some ChIP-seq, just to summarize data again, is that STAT1, which is downstream of type 1 interferon signaling, binds very strongly to the promoters of both complement factor B and to C3, and that these are therefore likely to be drivers of gene transcription here. And so to prove that conceptually, we took control cells, or sham infected them or infected them with SARS-CoV-2 in the absence or presence of Ruxolitinib. Ruxolitinib is a specific inhibitor of JAK1/2 STAT1 signaling. So we essentially blocked this signal. As you can see here, all of the complement genes that we're interested in were induced by infection, and all of them were normalized by Ruxolitinib. So complement factors in infected epithelial cells of the lung are driven by STAT1 to produce these factors. And what was particularly interesting was to show what happens on the protein level and how we can interfere here. So this is confocal staining. At the top, we have mock infected cells. So there's no SARS-CoV-2 here. This is Herc staining. This is SARS-CoV-2 N protein, and this is C3a, the breakdown product or the activation fragment of C3. As you can see, there is none of these present in uninfected cells. The moment, though, that you give the cell SARS-CoV-2 infection, you start seeing protein expression driven by the virus, and also the generation of activated fragments of C3a. As you can see here, here's the overlay. And in fact, those things are linearly correlated with each other. And what we managed to do was to collaborate with GSK and find a specific inhibitor of complement factor B, which is one of the enzymes that activates C3 in serum. And remember that we had shown previously that complement factor B is driven by C3, by STAT1. Well, here we showed that if you block complement factor B with a cell permeable inhibitor, you lose the ability of cells to generate C3A. So to summarize this story, and again, you can read all the details about it over here. The SARS-CoV-2 infects cells, and within the cell, it generates a type 1 interferon response. So interferons come out, they bind to the interferon receptor and signal through STAT1, where cooperatively with NF-kappa B, they lead to transcription of a bunch of complement genes. C3 here is processed by complement factor B, so 2 active fragments that go on and bind the cognate receptors on immune cells and drive inflammation in the lungs. And we could normalize this with a JAK-STAT inhibitor or with a complement inhibitor. And so this story came out again earlier on this year. You can read all the details of this later on if you're interested. But what I want to do is to use these 2 as illustrative cases to move on to the unpublished data, which I hope you'll find interesting. So remember that we said that, that there is a lot of complement in this tissue environment. So it's coming from here, it's coming from here. And what is unclear is how do these cells shut down, right? So what is the phenotype of these cells in tissues and how do they normally shut down? So we approach this in a similar way as we did before, focusing on transcriptional signatures within immune cells. So the summary at the end is that we found that this shutdown program is dependent on the interaction between vitamin D and a number of transcription factors that include BACH2, and all driven by intracellular processes within inflammatory cells. And I'll explain those things bit by bit. So let's talk about immune cells for a second. So generally speaking, naive T cells within an environment are induced to differentiate to a number of different fates. And these can be, broadly speaking, inflammatory or anti-inflammatory. And let's take an example. So IL-12 induces Th1 cells to express interferon gamma, and they upregulate their master transcription factors such as TBET. So based on this, the first question we asked is, what is the phenotype of T cells in infected lung biopsies of patients with COVID? So we turned to single-cell RNA sequencing that was publicly available at this point, and we identified the T cells, the lymphocytes within the lungs. And here is a cluster of cells that conform to CD4 positive T cells. And we're comparing here healthy versus COVID-19 lung biopsies. In fact, these are bronchoalveolar lavage fluid CD4 cells. So we looked at the expression of genes conforming to the different phenotypes or lineage commitments that T cells can undergo. And if you look at these and express them as a module score, you can see immediately that the genes that are part of the Th1 program are much more highly expressed in COVID-19 samples than in healthy controls, as opposed to very little differences in Th2 and Th17. So the lungs of patients with COVID-19 contains an environment in which T cells are skewed to the Th1 phenotype. And we know already that this is a complement-rich environment. So that being the case, we know, as we discussed earlier, that in this environment, C3 complement fragment protein is being processed to 2 components, C3A and C3B. And we've already said that C3b binds a surface receptor called CD4 T. Now, in the normal setting, this signature, a T cell receptor signal and CD4 T results in the following behavioral phenotype in T cells. So the first thing that happens is that cells start off not producing any inflammatory cytokines. Then they switch to interferon gamma production. This is your classic Th1 phenotype. If you leave them long enough, the cells will now start to produce IL-10. So these are now double positive for interferon gamma and IL-10. And if you leave them long enough, they will shut down interferon gamma and start producing only interleukin-10. So this is the normal fate decision or the life cycle of a Th1 cell that has been generated through complement signaling. And so if you take, so we asked the question, in these patients who have COVID, where are the T cells at in their life cycle? And unfortunately, IL-10 is below the detection threshold for most single cell RNA sequencing technologies at present. So instead, what we did was we went to bulk RNA sequencing of bronchoalveolar lavage fluid T cells. And what you can see is that this is a gene set enrichment analysis plot. So what we're looking for is Th1 genes. So let's say there are 250 of them. Each line down here represents one gene. And so where they are in this scale represents whether they are more highly expressed in cells from the patients or more highly expressed in cells from a healthy donor. And as you can see, they're all bunched up at this side. So what we can say is that genes more highly expressed in COVID are enriched in Th1 genes. So this is we've already shown at the single cell level. But if we now look at IL-10, what you can see is that the level of expression of IL-10 is about 8-fold or whatever lower in the patients that have COVID-19. So there is a dysregulation of the normal life cycle of a T cell. Instead of going to gamma, then switching down, they seem to be stuck in the gamma producing phase. So in order to gain some insights into some of these mechanisms and why things have gone wrong, we had to first figure out what the normal shutdown program of a T cell is. And so what we did was we used our lovely technology by sorting cells from quadrants A, B, C, and D and performing transcriptomic analysis. So I'm going to summarize a lot of data to say that this is essentially what we found, that there are a number of transcription factors that are induced during the life cycle of these T cells. And among these, among the very top was the vitamin D receptor. And so this was a real surprise to us. But when you think about it, when we thought about it, it kind of made sense, because if we just compare healthy donor to COVID lung samples, so lung tissue, all their CD4 cells, and try and calculate the upstream regulators, what we see is that the vitamin D receptor is predicted to be one of the top transcriptional regulators in both types of analysis. We further found, and this was really key, that one of the genes that is also highly induced in complement activated cells was CYP27B1. Now this is the 1-alpha hydroxylase that activates vitamin D. Now I'm going to come back to that in a second. So just to summarize this part, we confirmed that VDR and CYP27B1 are both induced by complement. And we did lots of different experiments, knockdown experiments, etc., natural knockout patients. This is the summary of vitamin D biosynthesis. So vitamin D is a secosteroid. Ultraviolet light, UVB specifically, from sunlight, breaks one of the chains of cholesterol in the skin, of 7-dehydrocholesterol to be exact. And this generates what we would call vitamin D. Now, vitamin D requires 2 hydroxylation steps in order to become biologically active. The first step is catalyzed in the liver and generates 25-hydroxyvitamin D. And the second step is catalyzed by the enzyme we just talked about, which is mostly expressed in the kidneys. At least, until a few years ago, it was thought to be exclusively expressed in the kidneys. And then what vitamin D does is that it ligates its receptor, which is the vitamin D receptor, and that translocates into the nucleus in cooperation with RXR. They bind vitamin D response elements, and that they generate the transcriptional response to vitamin D. And so what we saw and could summarize here was that T cells seem to be able to both fully activate and to respond to vitamin D, and that the complement receptor ligation induces this system. So this is novel. And it really made us wonder, well, okay, in that case, what exactly does the vitamin do in that case? What does vitamin D do in these cells? So again, we went to our usual approach, unbiased. We took some T cells, we activated them, and gave them either carrier or vitamin D in addition and did RNA-seq. And not surprisingly, lots of genes were differentially expressed. So let's say around 450 genes were up or down in vitamin D treated cells compared to carrier. And again, I'm going to summarize a bunch of data by highlighting the key findings. So this is a heat map showing expression of select genes. This is three donors, so no activation conditions or activation with carrier or with vitamin D. And so a group of genes were induced, which are known to be classic vitamin D induced genes. So CYP24A1, CTLA4, CD38. Well, this is good. This is a positive control. The next thing we found was that a bunch of cytokines were repressed. And these are classical inflammatory cytokines like interferon gamma, IL-17A and F. So vitamin D repressed these. We were a bit perplexed. We saw some cytokines that were induced, including interleukin-6. Now, isn't that a surprise, given that it's repressing all of these inflammatory cytokines? We also saw that a bunch of transcription factors were induced. So highlighted here are STAT3, c-Jun, and BACH2, and we're going to go through each of those in turn. But first off, let's look at these cytokines. So another way you can approach this data is to try and see whether there is a hierarchy here that helps you select one or two for further study. So what we did was we arranged all the genes that were induced or repressed by vitamin D by fold change. So the ones that were most induced are at the top, the ones that are most repressed are at the bottom. And in blue is every differentially expressed gene, and in orange is every differentially expressed cytokine. So as you can see at the bottom end, interferon gamma, IL-17, these are among the most repressed genes. That's good. On the other hand, we were really surprised that IL-6 is the most highly induced cytokine gene. And this isn't, a divide by zero. It's not that there was nothing there and then it goes up a little bit. There was a substantial increase. And IL-10 that I've highlighted here was number 3 or number 4 in terms of the induced cytokines. So this was quite interesting. Of course, what do? First of all, you don't believe it. And so we sorted and sorted and sorted like ultra pure T cells. And I can promise you the transcript is there, the protein is there, fine. So do they actually make much? Well, so this is protein concentrations in the supernatants of cells activated with vitamin D. So as you can see, interferon gamma, dose-response relationship with vitamin D, it's repressed. IL-10 is induced very nicely. And look at this, interleukin-6 is induced, but this pattern very closely mirrors that of IL-10. In fact, very rarely do you see this in biological systems, that the correlation between these two proteins has an R value of almost 1. When you see this, either both things are being regulated by an independent third factor in exact stoichiometry, or one of them is driving the other. So we, of course, did the next obvious experiment, which is to block signaling through the IL-6 receptor. So we repeated this experiment, but cultured the cells with either nothing or carrier or tocilizumab. And this is an inhibitor of IL-6 receptor. And what you can see is that the production of IL-10 in response to vitamin D is significantly blunted by blocking, as much as this drug can do, IL-6 receptor signaling. All right, well, if that's true, what does the structure of the IL-6 receptor tell us? So this is the IL-6 receptor. It's part of a family of cytokine receptors that shares a signaling chain, GP130, and it has a high affinity chain that's specific for IL-6. So when IL-6 binds here, GP130 signals through JAK-STATs specifically to recruit STAT3 to the nucleus. So if this is true, we should expect to see a change in the phosphoproteome of vitamin D treated cells, right? So what we did was we again took an unbiased approach and we did a phosphoproteome assay. So here we have these are phosphoproteome analyses of carrier-treated or vitamin D-treated lysate cells. So you take the lysates of these cells, you put them on this card, and you basically do a large Western blot. So what you have here, each 2 dots next to each other is a replicate. And you compare the intensity of these versus the intensity of a reference on both cards, and you normalize them against each other. So what you can see, for example, is there is really big increase in phosphorylation of c-Jun. There's a really big increase in phosphorylation of STAT3. So to summarize it as a heat map, these are the changes in the phosphoproteome that were statistically significant. And so, of course, STAT3 and c-Jun are the ones that really stood out. And if you remember, this is important because in our transcriptional data, we had shown that both STAT3 and c-Jun are transcription factors whose gene transcription is induced by vitamin D. So again, now that we've done the sort of the big data stuff, we can do a very simple experiment in the lab. And what we did was to do a Western blot of vitamin D treated here or carrier treated cells. And so what you can see is if we look at STAT3, total STAT3 levels go up with vitamin D treatment. That's good. We see that in the transcriptional data as well. But phosphorylation of STAT3 also goes up really significantly. And if we repeat this experiment, where we block the IL-6 receptor with tocilizumab, what we see is that we lose phosphorylation of STAT3, but not protein levels of STAT3. That means that STAT3 transcription and translation is dependent on vitamin D, but phosphorylation of STAT3 is dependent on IL-6. And in terms of c-Jun, we see a big increase in c-Jun and in phospho-c-Jun, but this is independent of IL-6 receptor signaling. So we did a number of experiments that confirmed that we're correct on this. So we saw that IL-2 that's induced by vitamin D is impaired if you use chemical inhibitors of STAT3, if you use siRNA knockdown of STAT3, and of course, if you take cells from patients who have dominant negative mutations of STAT3. So we're very convinced that this is correct, that vitamin D induces the expression of a set of cytokine genes and transcription factors. These include IL-6, STAT3, and IL-10. And we showed that IL-6 drives IL-10 production by phosphorylation of STAT3. All right. Now, how does vitamin D shape gene expression then? Okay. So let's look at some basic examples of genome-wide architecture. So here is a gene over here. Let's say this is gene Z. This gene has a very rudimentary structure. The entire transcriptional output from this gene is dependent purely on its promoter. A gene up here, gene X, has a regulatory region, which let's call it an enhancer, sort of in close proximity to it. And the activity of the enhancer alters transcriptional output from the promoter. There is another kind of architecture over here in gene Y, which is a particularly complex area containing multiple enhancers, which for the sake of argument, we'll call a super enhancer. That means that this gene is much more intricately regulated. Now, our view of enhancer-promoter interactions is shaped by how we believe the DNA to fold. So in general, a model that explains most observations is that DNA folds to bring enhancer and promoters in close proximity to each other, and that there's an interaction between these two that is mediated by a number of transcription factors, and that recruit POL2 for gene transcription. Now, why this model is great is that there are specific markers for promoters and enhancers for which we have good antibodies. So for example, on the enhancers, we have H3K27 acetylation. So histone 3, lysine 27 acetylation. And what this means is that we can actually probe for these areas to find the active areas of the genome. And that's usually done by ChIP-seq. Now, the problem with ChIP is that you need large cell numbers. This is hard to obtain if you're ever going to do stuff with patients. You need ChIP-grade antibodies. You have to cross-link the cells, and that introduces artifacts. It's very operator and sonicator dependent. And I can say that most people, even if they're good, on a day-to-day basis, will have only about 50% agreement between their different samples. And if you're using bulk populations, then you have some difficulty with graded signaling. So I think it's fair to say that we needed a better method. And around the time when we were considering what we should do, our colleagues from Washington State described a new technology called CUT&RUN. So just to take you very briefly through this, you take a cell or nucleus and you bind it to con A. And so this stops the nucleus from floating away, or the cell from floating away. And you're trying to find this acetylation locus here. So you permeabilize the cell, and incubated with an antibody to your histone modification of choice, having first attached an emanase to this particular antibody. So let's say you use PAG emanase, right? So incubating these together brings the antibody and the emanase right into proximity with that acetylation region. And this just sits there until you add calcium. And that activates the enzyme of the emanase, the enzymatic, the catalytic domain of the emanase. And it digests the DNA, freeing that complex. And what you then do is you purify the DNA from that, you sequence it, and you get a very high signal to noise ratio. So the advantages of cut-and-run over ChIP-seq is you don't need very many cells. So we've done it, I think the lowest we've done is 500. It's very high resolution, so your peaks look really different. You don't need very many reads, has a high signal to noise ratio, and it's not so operator dependent. It's much more reproducible. All right, so we did exactly that. We looked for H3K27 acetylation in the context of vitamin D. And what we found was really quite remarkable, that within 45 minutes of getting vitamin D, there was really quite significant architectural rearrangement in the enhancers of T cells. A better way to show this is the differential H3K27 acetylation peaks. So you can see all of these peaks that are suddenly induced and the changes that happen over time. So this is dynamic epigenetic reorganization induced by a vitamin hormone. It's really quite remarkable and fascinating. All right. So I'll show this to you graphically. It looks like this. So here are all the peaks that are repressed by vitamin D. This is just a heat map, right? So this is the carrier treated cells, and this is the vitamin D treated cells. You can really see the repression of these marks. And that's summarized here by the histogram. So in red is carrier and in blue is vitamin D. What you can see here is the reverse. So these are the vitamin D induced acetylation peaks. As you can see, it substantially increases. There's another way to look at this, which is to subtract the two from each other and organize them hierarchically. So this is a hockey stick or a shoulder plot, if you like. So this is a way of ranking the size of the enhancers. And in general, anything that's above this point here, where there is a tangent to the curve with an R value of 1, are the domain annotated as having super enhancers. So what you can see here is that the top gene is BACH2, right? So now we have genome-wide reorganization and the development or enlargement of super enhancers. And among these genes here, we also have CTLA4, CD38. Remember, these are classical vitamin D-induced genes. We have IL-10 and STAT3 again. Wonderful stuff. And to illustrate what this looks like in a track, this is a genome browser track. So this is histone acetylation marks in carrier-treated cells around the BACH2 locus. And this is what happens in vitamin D-treated cells. You can see all of this acetylation deposition around this region. And this whole part is probably one really large super enhancer. And we're going to come back to this diagram and build it level by level. All right. So now we have a map of changes in the epigenome of T cells mediated by vitamin D. All right. So the second thing we can do is say, okay, now we have all of these. What are the transcription factors that are binding there? So we can approach this like this. Let's take the frequency of the motifs of transcription factors underlying those peaks and compare them to, say, the frequency of the same transcription factor DNA motifs in random regions of the genome with similar size. That's not quite how it's done, but let's say that's how it's done. And this gives us a score for the motif enrichment of transcription factors and helps narrow down the search space for the genes that you're looking for the transcriptional regulators that you're looking for. And I'm going to summarize it here again in a heat map. So this is the time point. This is the transcription family, transcription factor family. And over here are the representative members. So the top transcription factors are AP1, and there we have it, boom, BACH2 and c-Jun. We have VDR and we have STAT3 among the top transcriptional regulators whose motifs are represented. That means the fingerprints of these transcription factors are within vitamin D treated cells. So this is important because in our transcriptional data, if you remember, STAT3, c-Jun, BACH2, all three induced by vitamin D. So they're induced transcriptionally and the fingerprint is there. All right. This is now cut-and-run and cut-and-tag, a related technology for those transcription factors. And just look at these heat maps. This is the intensity of binding of c-Jun, VDR, STAT3, and BACH2 in carrier-treated versus vitamin D-treated cells genome-wide. I mean, you can really see the increase in binding. So the transcription factors go up, the fingerprint is there, and there's genome-wide binding. And down here, we're focusing only on the areas in which vitamin D induced more acetylation. So what you can see is that in vitamin D treated cells, these transcription factors are recruited to the new enhancer and super enhancer loci that have been generated. All right. Now, the second thing we can do based on our cut-and-run and cut-and-tag data is to assign gene regulation to bound transcription factors. And so what we can say is that of all of the genes that are regulated by vitamin D, 60% of them are bound by at least one of these transcription factors. Many of them are bound by 2, 3, or 4. So that means that we can explain up to 60% of the variance of gene expression induced by vitamin D based on 4 transcription factors. And a way to look at it as a heat map graphically is like this. So carrier-treated cells, vitamin D-treated cells, and these are the transcription factors that bind those loci. And over here we have our friends, STAT3, IL-10, BACH2, CTLA4, etc. There's another way to look at this, which is to construct a network diagram. And that looks like this. So this is the regulome of vitamin D treated T cells. So the majority of genes have c-Jun binding. These are the genes that are BACH2 bound, the genes that are STAT3 bound, and the genes that are VDR bound. And in the outer corner, we have each of the genes and the changes of expression induced by vitamin D color-coded according to this heat map over here. So this is what vitamin D does to regulate genes. Now, we are particularly interested in BACH2 because BACH2 is one of the most important transcriptional regulators that we study in Tregs. So I'll show you an example track of BACH2. So we've already seen this part here. This is the H3K27 acetylation locus tracks for vitamin D and carrier. And this is what the rest looks like. So the order is the same, carrier in red, vitamin D in blue. And what I've done is to put a dot underneath each of the new peaks that are generated by vitamin D. So here's the super enhancer region. These are all new peaks in c-Jun, new peaks by BACH2, new peaks of STAT3, and new peaks of VDR. So what's happening is that this region is developing the super enhancer and it's recruiting these 4 transcription factors to hopefully transcribe the BACH2 gene. Now, is BACH2 itself actually induced by vitamin D? So remember, this is the transcription data. So yes, it's transcriptionally induced. This then is a Western blot that we did. So here is BACH2 in carrier and vitamin D treated cells at 48 and 72 hours. And of course, the protein accumulates, which is good. Here is in vivo data. So here we took skin of patients who had psoriasis and either gave them no treatment or vitamin D treatment, the patients, and then took the skin, I should say. The reason for psoriasis is because they have T cells in the skin. Normal skin doesn't have very many T cells. And this is confocal imaging, looking for CD3, BACH2, and DAPI. So just focus, I've only blown up quadrants 1 in each of these. So here is no treatment. You see lots of CD3 and no BACH2. And down here, you can see the cells, the skin that has received vitamin D is now producing lots of BACH2. And more importantly, you can't see it here, it's Z-stacked. A lot of this is within the nucleus. And these have the appearance of PML bodies, which is one of the mechanisms that BACH2 regulates genes. All right. So how important is BACH2 for the regulation of vitamin D-induced genes? So if you take all genes in the genome, 5% of them are bound by BACH2, but 15% of vitamin D regulated genes are bound by this transcription factor, which is a big enrichment. And so we can also look at this by gene set enrichment analysis, as we did before. And what we can say is that BACH2 bound, BACH2 induced genes are more highly expressed in vitamin D treated cells. And BACH2 bound, BACH2 repressed genes are more highly expressed in carrier treated cells. That means that there is a BACH2 signature within vitamin D treated cells. I'm going to draw your attention here for 1 second to the IL-6 receptor. Right? This is among the most highly enriched genes in vitamin D treated cells. We'll come back to that. Now, to prove this, we took cells from a patient who has a haploinsufficiency of BACH2. This is a patient, a disease that we described in 2017, mediated by a mutation that causes protein misfolding and haploinsufficiency. So we gave cells from the healthy control and the patient vitamin D, and we looked at vitamin D induced genes. As you can see, these genes are significantly enriched in wild type cells, meaning that the absence of a full complement of BACH2 skews the transcriptional response of vitamin D. Another way to graphically represent this is with a Sankey diagram. This is a Sankey diagram. So all the differentially expressed genes are in the middle, and I've annotated some of them. So if you want to look at IL-6 receptor, for example, here. In red, red and orange, we have vitamin D induced genes, and in blue and sky blue, we have vitamin D repressed genes. And then we have the genotype. So this is the wild type, this is the mutant, wild type and mutant. And as you can see, the range of genes regulated by wild type is substantially different from the mutant. And over here on the right, we have annotation of the transcription factors that bound those genes. So having even half the amount of BACH2 radically changes the response to vitamin D, which means that BACH2 is obviously critically important here. So what does BACH2 do in all of this? I'm just going to simplify and show just one diagram very quickly. So down here is where the magic is happening. As you can see, IL-10 is not being induced in BACH2 haploinsufficient cells. And the reason for that is because the IL-6 receptor is not being expressed. If you remember, IL-6 binds to the receptor to transduce a STAT3 signal. And so this is a really good example. This is from mouse. This is a BACH2 ChIP-seq data over here. You can see lots of BACH2 binding to the IL-6 receptor. And this is BACH2 knockout versus BACH2 wild type RNA sequencing. As you can see, there's substantial decrease in BACH2 in expression of the IL-6 receptor in the absence of BACH2. And so what we think is happening is that BACH2 sensitizes cells to the effects of IL-6, and that's why you don't see IL-10. So I'm going to summarize the mechanism in healthy CD4 cells here. So what we've shown is that C3b binds CD4 T6, and together with a T cell receptor signal, it generates Th1 cells. So they make interferon gamma. At the same time, there is transcription of CYP27B1 and the vitamin D receptor. Now, CYP27B1 is the 1-alpha hydroxylase that activates 25-hydroxyvitamin D to 1,25-hydroxyvitamin D, which is a fully active form of vitamin D. This binds VDR, and that translocates to the nucleus. This complex now recruits a bunch of histone acetyl transferases and other chromatin modifiers to change the epigenome of cells, specifically around typical and super enhancers. The effect is to repress gamma and 17 and to induce CUT&RUN, BACH2, STAT3, and IL-6. IL-6 is secreted, binds the IL-6 receptor, that phosphorylates STAT3, which translocates to the nucleus, binds IL-10, and you make IL-10. And one of the roles of BACH2 here is to sensitize the cells to IL-6 signaling by transcribing the IL-6 receptor. So this is what happens when a cell gets a complement signal, and that's the mechanism for going from pro-inflammatory to anti-inflammatory. All right, back to the basics. So what happens in COVID? So remember that there is a very strong epidemiological association between the incidence and mortality of severe COVID and vitamin D insufficiency or deficiency. And there's lots of data for this now. I should really replace this slide with newer stuff. But what you can see is this is across lots of different countries. And this is the COVID-19 incidence and vitamin D levels. You can see that. This is mortality from COVID-19 and vitamin D levels. So the lower your vitamin D level, the higher your mortality. And there are numerous publications. And some of these are out in press. And there's a very large meta-analysis now out as well. So there is this association, but what about vitamin D repressed genes? So vitamin D, of course, remember, represses the Th1 program. And what we found is that if we look at these repressed genes in healthy versus COVID-19, those genes are de-repressed in patients who have COVID-19. This is single cell data from the CD4 cells in the lungs, remember. And what is particularly interesting is that if we look at the Th1 mess and the vitamin D repressed genes, there is this direct correlation between them. So that exists both in healthy individuals that down here and in COVID-19 up here. The idea then came to us that maybe what could happen is that there is a deficiency of either vitamin D or its mechanism of action is disrupted. And so we could potentially push cells down this way if we give them vitamin D or redress this balance. So how good is that vitamin D repressed module for distinguishing patient from control? Well, so here is the receptor operator curve and the AUCs for a Th1 score to distinguish patient from control. As you can see, this performs really well and the AUC is 0.9. The vitamin D repressed module actually performs really well too. The AUC is 0.8. To contextualize this, we took every single gene set that we could find in MSigDB, so there's 2,000 of them, and we did the same analysis and saw how well they performed. And so this is the rank. The best performing is, of course, it's an infection, so type 1 interferon, type 2 interferon responses. But the vitamin D repressed module is in the top 0.5% of all gene sets out of 2,000. So it's a very strong biomarker for COVID. Now, independently, we took the gene sets, the differentially expressed genes in the lungs, and we computationally predicted drugs that could normalize the abnormality. So these are the top 10 drugs. As you can see, 5 of them are steroids. This is good because we know that in the trials, steroids work. 2 out of the 10 are vitamin D analogs in this. So the computational prediction is also that vitamin D replacement or treatment is going to be useful, possibly in combination with steroids because the two synergize together. So the idea is this: the COVID-19 program is stuck somewhere here in the inflammatory phase. And if we give vitamin D or steroids, we'll be able to accelerate resolution towards an anti-inflammatory phenotype. And so this paper, in fact, half of the data is not in this bioRxiv submission. It's still under review. We've just submitted the revisions. The main people here are Daniel, Charles, Thilo, and Ruben, and Bing Yu that you've seen before, and Majid Kazemian, my great collaborator from Purdue. I'm going to stop there and thank everybody. There's lots of people involved here, particularly from my lab, from the complement lab, from the fungal pathogenesis section in NIH. And in particular, Majid Kazemian, with whom I do most of my work. He's a computational biologist and obviously essential to our work. Christiana Wobus is a key collaborator on COVID work over at University of Michigan. So for anyone here who knows anyone that might be interested in the kind of stuff that we do have postdoc positions available. And so I'd encourage people to apply if you're interested in this kind of work. And yeah, so please get in touch. And I'd be delighted to take some questions.
Okay, thank you so much, Ben. That was crystal clear. It's really refreshing. I think, Shelley, go ahead.
Hey, Ben, this is really a beautiful talk, very inspiring. So I think I have 2 questions. The first is about vitamin D. So the vitamin D pill like we got from Walgreens, So do you think that is sufficient to like change the transcription profile in T cells?
That's a really good question. So vitamin D, there's a number of things about vitamin D. So the first is that there's evidence that vitamin D that you make in your skin is much more effective than vitamin D that you take as a tablet.
Mm-hmm.
And part of that is because the vitamin D that you take in a tablet form has very variable bioavailability. So not all of it gets absorbed, or too much gets absorbed, and it has to first be activated in your liver. So a number of different factors are at play there. The one trial that I thought was particularly impressive with vitamin D in COVID showed that if you give an analog of vitamin D, one particular formulation that has a really high absorption and doesn't need liver activation, the chances of ending up on ICU was only about 10% of not having it. That was really impressive.
Yes, that's really impressive.
That's really impressive. I actually have been advising people not to go and take vitamin D willy-nilly, because some of the formulations of vitamin D have really high levels. So you could run into toxicity.
Mm-hmm.
Go out, enjoy the sun. 30 minutes of direct sunlight will replenish your vitamin D levels.
Okay.
Obviously, don't get sunburnt, but you know, go and enjoy the sun. That's a better way of doing it, I think.
Okay, so that's a much better solution.
Yeah, actually, I'll be honest with you, Shally. So I put all this paper together. You know, it was all great. Then I had a sort of a yearly appointment with my family practitioner. I went there and she did some blood tests and said, "Do you want your vitamin D levels measured?" I said, "Sure." So this was during lockdown. My vitamin D levels were undetectable because I had been indoors the whole time. You know, I just sent this paper out, so I felt a bit ashamed of that. But there you go.
Well, that's fun. Okay, so my other question is about the first part. So you talk about the integrin signaling induced the C3 in the T cells.
Yeah.
So is that dependent on the TCR signaling, or it's only like dependent on the integrin?
Yeah, no, you need TCR. So it's CD3 plus AP1. Now that's in vitro. Most T cells that are being recruited into those sites are already activated somewhere. So they have lower requirement for TCR signaling. Once they get into the cell, though, into the tissue, they will recognize cognate antigen in the context of APC. So they will get an additional TCR signal there.
I see. So the function of integrin here is that to like change the threshold to induce the C3, or it's more like a priming?
Yeah, really good question. So we didn't actually establish that one way or another. So what we should have done was to titrate up the strength of the signals. But the experiments actually were quite complex in vitro. And so what we showed was definitely a yes. What we saw was that we ended up calling it C3 licensing. So that signal, the AP1 signal, allows gene transcription of C3, and we think we know where in C3 the AP1 binds. And at the time, the technology that was available to us didn't let us get a good ChIP-seq done. So we were in a bit of a rush to send the paper back. So we didn't do the cut and tags of it, but we're pretty certain we know where it's binding.
Yeah. Okay, cool. Yeah, thanks so much.
Sure.
Okay, so I think the other person, Bing-Ching, you want to ask a question?
Oh, yes.
Go ahead.
Hello, Ben. Thank you very much. And I learned a lot from your talk. It's very wonderful. And I wonder that if IL-6 and vitamin D can induce IL-10, so how could we distinguish the inflammatory IL-6 or the IL-6 that are going to stop inflammation and induce IL-10? So it's very hard to distinguish, you know. Yeah. So especially during COVID-19, previously we suggest to use the tocilizumab to inhibit IL-6 receptor, you know. And now maybe we just maybe give them vitamin D to push the IL-6 to induce IL-10. So the time and, you know, we are very hard to distinguish two of them. So what's your comment?
Yeah, so I'm really glad you picked up on that. So two things. So in the paper, we actually show this. If you give IL-6 to T cells, they start making IL-17, as we all know, right? So, there's a bit of TGF-beta in the serum, so the IL-6 will induce IL-17. If you give IL-6 together with vitamin D, it does the opposite. So 17 is shut down and it starts making IL-10. So you have two options. One is, as you say, IL-6 receptor blockade to cut down some of the inflammatory functions of IL-6, or to give vitamin D, which will convert some of the inflammatory function to anti-inflammatory. Or you could combine the two together. Absolutely.
Yes, I think maybe we can give them two together is a better choice, right?
Yeah, so one of the real complexities about both of those things is that IL-6 and vitamin D are both very pleiotropic. So they perform different functions on different cells, and they are both cell and context specific, right? So vitamin D, for example, is also very important to generate some antimicrobial peptides, cathelicidins, beta defensins, for example. So it has some pro-inflammatory function on the right cells at the right time. I suspect it's similar to IL-6. In fact, most things in the immune system, right? Too much for too long is bad. Too little for too long is bad. So you need, it's like Goldilocks, you need just the right amount.
Okay, thank you very much.
My pleasure.
So, Ben, I have a lot of questions, actually.
Go for it.
First of all, actually, I'm curious about the complement. I mean, since you observe all the high-ranking signaling pathway, is that inflammation type dependent?
Is it what dependent?
What kind of, I mean, you're talking about virus infection, but I'm curious that what about the other type of inflammation? I mean, that's the reason why you see it's highly, yeah.
I love this question. So until about 10 years ago, people thought that complement is exclusively made by the liver and secreted into the blood. Then around 7 years ago, I was part of a group that demonstrated that complement exists intracellularly in T cells. And that was the hardest paper we've ever tried to get published. So far, we found complement in virtually every cell type that we look for. So I'm right now working on another paper showing it being produced by kidney cells. And probably what it does is that it has very, very important functions for basic physiological processes of cells. So in T cells, for example, it's incredibly important for activation of mTOR and for nutrient channels to allow T cells to become Th1s, to alter their bioenergetics. In synovial fibroblasts, a really nice immunity paper has recently shown that they are important for response to sterile inflammation. So not crystals. Yes, uric acid crystals. And in fact, it helps reshape the epigenome of synovial fibroblasts. So they develop some kind of memory response. And so on re-challenge, they get much more severe inflammation, and that's earlier and more pronounced. There's a story that we're putting together right now about the role of complement, intracellular complement in cardiovascular disease. So it's going through a renaissance right now. People used to think, oh, we know everything about complement, but it's really going through a renaissance. And there are people that have been in touch with us that are working on it in the context of the brain and neurological function. So I think you'll find a profusion of complement papers. I mean, we've had what, 3, 4 in the last few years. There's, I've got another one I'm working on, a second one, sort of a year from now, I think. It's highly relevant.
But I guess my question is because your screening system is basically based on different type of extrinsic stimulation. I mean, let's say viral infection. My question is, the reason you see it's highly, if I understand this correctly, you're talking about even in the physiological condition, this actually signaling pathway is highly activated. That's what you're saying?
there's small amounts of C3 expressed all the time in T cells.
Right.
That's required for basal mTOR signaling. If you knock that down, the T cell dies, actually. It's really interesting.
Sure. But
In the respiratory epithelial cells, a number of stresses can induce it, not just infection. And by the way, it's not specific for SARS-CoV-2. So another group showed a couple of years back that serum starvation, so if you stress epithelial cells, they start making C3. And in that scenario, the C3 was protective. So my suspicion is that a lot of different cells have the ability to make C3. And up to a certain level, it's protective. If you have too much of it for too long, it becomes pathogenic by recruiting inflammatory cells, changing bioenergetics of cells, etc.
Okay, I see. So my second question is, I mean, you're showing this nice, destination of different T cell evolved during the process of inflammation. So when they actually start to from the double negative to interferon gamma positive and then eventually become IL-10 positive, is there actually a evolution or evolved between the Th1 to Treg cells?
You almost had the right question. You almost had the right question is, what is the relationship of these cells to TR1 cells, right? So they're FOXP3 negative, right? So they can't be your traditional Tregs, but they can be TR1 cells. So FOXP3 negative, gamma negative, IL-10 positive. So this is going to sound really controversial. The transcriptome of these cells is indistinguishable from sorted TR1 cells. So my suspicion...
Compare with Th1.
TR1.
No, TR1 compare indistinguished from the Th1.
Both.
Okay.
Th1 and TR1 cells and shut down Th1s are indistinguishable from each other transcriptionally. And so I think what we'll find is that every T cell has the capacity to shut down its own inflammatory program, and most of them do so by making IL-10. So probably what we had previously defined as IL-10 producing regulatory cells, TR1 cells, probably don't exist. These are shut down T cells, inflammatory cells. That's my guess. I mean, others can do that work.
I see. Okay.
I'm going to do other stuff.
The third question is, actually, I'm following the previous one. So, so you're saying that when you actually have extra vitamin D, then you see even with the presence of IL-6, if there's no vitamin D, then it's become Th17 cells. But when you have it, it's repressed. Can you comment on that? What's the potential machinery?
Mechanism? Oh, the mechanism is epigenetic reorganization.
For the
So the loci pertaining to IL-17 and interferon gamma are silenced. So H3K27 acetylation goes down at those loci. How does it do it? So vitamin D recruits a number of transcriptional epigenetic modifiers. I concentrated here on the histone acetyl transferases, but it also recruits repressive elements to it. What we didn't do, we could have done, was to do CHIP for some of those as well. EZH2 and others. We just didn't. There were, these are quite a lot of experiments already. So,
but without IL-6 present, can you still see vitamin D alone can actually epigenetically modify these loci like IL-17?
Yeah.
Can you still see that?
Yeah. So vitamin D does all of this on its own. The only way because the cells make their own IL-6, the only way to do it in the absence of IL-6 is vitamin D plus blockade of the IL-6 receptor, or CRISPR out IL-6 or the receptor, and do that. And we didn't do that part. I mean, we assume that the two are linked.
Okay. I think my last part is, I'm curious about the last part. You're talking about the BACH2 mutant patient, the patient. So because I assume that even in the patient cells, the vitamin D still can induce BACH2, right? But somehow you see completely different landscape of the binding. I mean, can you comment on that?
Yeah, so BACH2 has a really big super enhancer. That means that most genes that have really large super enhancers, so this is a bit of a generalization, but tend to have a linear response in terms of their dose-response relationship with gene expression, right? So this particular patient only has one functional BACH2 protein. So although transcriptionally, BACH2 levels go up, so the transcript for BACH2 from both alleles goes up, one allele produces a mutant protein which is degraded. It has a mutation in it. We did some really I really sort of
I read the paper. I read your BACH2 paper. Yeah.
Yeah. So I love that part. So the protein doesn't fold and it breaks down. If we had really pushed that experiment, we could probably have uncovered that the mutant protein also has a small double dominant negative effect as well. But most of it is simple misfolding. So you only ever get about 50% of the total protein that you would do with normal expression.
But transcriptionally, the vitamin D can equivalently induce BACH to mutate gene as the Y type.
Yes. Transcription goes up, but not translation.
Right. But my question, actually, I think that's you should know. I mean, the BACH2 should be self-promoted or not. It's by its own promoter.
BACH2 binds its own promoter, correct,
Okay.
and it induces its own transcription. Yeah.
Okay. I see.
Bit like Jun. Yeah.
Yeah. Okay. Perfect.
A lot of AP1 proteins are like that.
Yeah. Okay. So I think if anyone else doesn't have questions, I'm sure Ben happy to take any question by emailing him, right, Ben? So if they have questions or you want to interested in join Ben's lab, I think he's also very welcome. But thank you so much again, Ben, for the wonderful talk. I mean, I'm learning so much. Really grateful for this wonderful seminar. So hopefully everyone else, I see you next time. So good night, Ben. Thank you so much.
Thank you very much. Have a good night, everybody, and good morning for the ones.
Good night. Yeah. Bye-bye.
Bye now.